So part 2 of the Unit 1 lecture, we're going to start off with some taxonomy and classification information. So taxonomy is a consistent worldwide means of classifying, naming, and identifying organisms. Organization of biological species into categories based on relatedness. So what similarities do they have? And based on these similarities, how can we group them? Most bacteria used to be grouped by physical characteristics, but now they're more often grouped by DNA and RNA, and therefore species are often changing when new information is gathered. So a lot of the time, if you have an update to a microbiology textbook, it's going to be because classification names have changed. So one way that we can classify organisms is by nomenclature. So taxa or categories include the domain, which means are they a prokaryote or a eukaryote, the kingdom, the division, which is the phylum used in the kingdom, class, order, family. So the family is indicated by that ACA ending on the word. Tribe, genus, species, serological groups, and strains. So, and if you've taken any biology course previously, those categories probably sound familiar to you. The only things that we really need to worry about for the purposes of this class are the family, genus, and species. Then some organisms are further divided into subspecies. So an example of the family, you can kind of think of that as like the clan or the larger group that the organism belongs to. And again, those are the ones that will have that ACA ending on their name. So an example of that is the Micrococcaceae. The genus, you can think of this as the organism's first name, should be capitalized always and italicized when in print. So an example of that would be Staphylococcus is the genus for Staphylococcus aureus. And then the species is the aureus. So you can think of that as the organism's last name. It will be in lowercase, and it will also be italicized when you see it in print. So a different way that we can classify organisms is by their cellular type. So are they eukaryotic cells, which means that they have a well-defined nucleus and contain organelles within the cell. So examples would be fungi, algae, animals, and plants. Or are they prokaryotic? So the word prokaryotic means before nucleus. So they're going to have a poorly defined nuclear membrane, no organelles. This is the category that bacteria fall into. And then archaeobacteria are more closely related to eukaryotic cells than to prokaryotic cells. Their cell walls lack peptidoglycan, which is a major reason why they are placed in a separate domain from bacteria. But like bacteria, they can stain gram-positive and gram-negative. So this includes microorganisms that grow under extreme environmental conditions. They're able to grow in so those that are known as able to grow in extreme environmental conditions. An example of that would be halophiles, which means salt-loving, so those organisms that are able to live in the Great Salt Lakes. Another way that we can classify organisms is by their cell wall characteristics. So in prokaryotes, the cell wall is rigid in order to maintain the shape of the cell, and it helps to prevent bursting of the cell. There are different types of bacterial cell wall structures. So we've mentioned a couple times previously gram-positive and gram-negative cell walls based on how the organism stains with the Gram's stain. So gram-positive organisms have cell walls that are made of a very thick protective peptidoglycan layer with teichoic acid crosslinks. Gram-negative organisms have cell walls that are composed of two layers, an inner peptidoglycan layer that is much thinner than the one in gram-positive organisms, and then also an outer membrane that contains proteins, phospholipids, a lipopolysaccharide that functions as the cell's initial barrier to the environment. And then the last way that we can categorize our organisms is by DNA or RNA relatedness. So as technology develops, we are able to tell more about bacteria and classify them on the basis of DNA and RNA relatedness rather than just their morphological or biochemical characteristics. Here's kind of an overview. I'm sure, like I said, if you've taken a biology course previously, this is just kind of like that taxonomy structure. Okay, so next we're going to talk about some morphology and different identifying characteristics. So the different microscopic shapes, bacteria occur in 3 shapes: cocci, bacilli, and pleomorphic. So cocci are spherical. They may occur singly, so either by themselves or in pairs. If they're in pairs, they're referred to as diplococci. They can occur in chains, which is evidence of a strep species, or they can occur in clusters, which is evidence of a staph species. And this is one of the main reasons why that initial preliminary report, either the wet mount or the gram stain, can give information to the doctor so that they can begin treatment. Bacilli are rod-shaped. They can vary greatly in size and length, from short, football-shaped, which are called coccobacilli, to long filamentous rods. The ends may be square or rounded. Bacilli with tapered or pointed ends are called fusiform. Vibrio are a gently curved rod, so you can see him there. Spirillum are slightly curled or spiral shaped. So you kind of think of a corkscrew. And spirochetes are spirals also, but they're more flexible, so they kind of resemble a spring. All of those are considered rods. They're just different morphological variations. And then pleomorphic is not a bacterial shape necessarily, but a term that refers to a species that can vary in size and shape within a pure culture. Okay, with the Graham's stain, Graham's is the most fundamental test that is used for bacterial classification. So nearly all clinically important bacteria can be detected using this method and are classified as one of 2 general types, either gram-positive or gram-negative. So the purpose of this is primary microscopic examination of the specimen submitted for culture. So it can be an indication of infection and lead the physician in initial treatment. So like we talked about in the first video, when we do this preliminary gram stain, we can have that result out generally within about 30 minutes. The culture, once we've inoculated the media, it has to incubate. It might be 12 to 16 hours before we can put out a preliminary report based on the growth from the media. So these gram stain reports are very important. Reagents involved in the gram stain: crystal violet is the primary stain. So the slide will go into the primary stain for 1 minute. It enters the cell, binds to that thick cell wall of gram-positive organisms, so that thick peptidoglycan cell wall that has those teichoic acid crosslinks. Then it goes into the iodine, which acts as a mordant or a fixative. It goes in there also for 1 minute. That helps to bind that primary crystal violet stain to the bacteria. Then it goes into a decolorizer, which is an acid alcohol, for 10 seconds and then a quick rinse. So it will remove the crystal violet from gram-negative organisms, which have the much thinner cell wall. So gram-positive organisms are not as thin. They won't be penetrated by that decolorizer, so they will hang on to that primary crystal violet stain. And then lastly, safranin, which is a counter stain. The slide will go into the safranin for 30 seconds. So gram-negatives that have that much thinner cell wall, that have had the crystal violet removed by the decolorizer, they will be stained with safranin. So gram-negatives take up this stain because their cell walls have a high lipid content, a thinner peptidoglycan layer. You also have to remember, before staining, the slide will have to be heat or methanol fixed. So once you apply the organism or the specimen to the microscopic slide, you have to wait for it to dry. Then that specimen has to be fixed to the slide, so either with heat or with methanol, and that just prevents the specimen from coming off of the slide while it's being stained. So you can see here, this represents a gram-negative organism because it is stained pink. So safranin is a pink, that's our secondary stain. So this is a gram-negative bacilli because it's a long rod-shaped organism. And then here we have gram-positive cocci. So it's gram-positive because it's purple, it's retained the primary crystal violet stain, and then it's classified as a cocci because it's round or spherical. And then here again is just a kind of a run through of what's happening during that gram staining process. So you start out with an organism that enters into the crystal violet stain. Initially, both gram-positive and gram-negative organisms are going to take up that primary crystal violet stain. Then they go into the iodine that helps fix that crystal violet into the cell wall of gram-positive organisms. But once it goes into the decolorizer, because the gram-negative organisms have that much thinner cell wall, it will wash away that primary crystal violet stain. And then the last well is the safranin or the counter stain. So those organisms that are gram-positive have held on to that primary crystal violet stain the whole time, whereas the gram-negative cell walls will now pick up the safranin. This is just a picture of an automated stainer. So in class, you guys will be practicing manual staining techniques, but a lot of labs have automated gram stainers where they have an arm where you can load a bunch of different microscopic slides at one time, and then each one of these wells has the stain loaded into it. So the arm takes the slides and dips it into the well for the appropriate amount of time. And this is just a much more consistent method of staining. So as I mentioned, let me go back up a couple slides. The results from your gram stain can be either positive, which is purple. You'll see purple when you're looking at the organism under the microscope. So it retains that primary stain. Every test that we perform in the lab, we have to do positive and negative controls on. So this is to ensure that the testing that we're doing is accurate, that our reagents are working properly, and that we can feel confident when we report out our patient results. So Staph aureus is the control organism for our gram-positive organism, or for our gram-positive stain. So gram-positive organisms have cell walls made up of peptidoglycan and teichoic acid cross-links. Our gram-negative are, again, they retain that secondary stain, the safranin. E. coli, the organism E. coli, is our control for the gram-negative. And remember, gram-negative organisms have cell walls made of lipopolysaccharides and peptidoglycan. It's a much thinner cell wall. And then some organisms can be variable. So they're known as gram variable. You will see pink and purple on the slide. So some other morphological structures that can be seen on the gram stains that can help us with identifying organisms. A capsule, so these are different cellular structures. So a capsule is a covering. It sometimes gets referred to as a slime layer. Usually does not stain. So what you'll see on the microscopic slide when you're looking at it is kind of a halo around the bacteria on the gram stain. This is a type of virulence factor, so which means that it's something that makes the organism more virulent. So this can be made up of polysaccharides or polypeptides. They're going to provide defense against phagocytosis. So that in itself is a virulence factor. Phagocytosis is our body's way, our cells in our body, what they do to identify and get rid of pathogens in our body. And it facilitates and maintains bacterial colonization of biologic and inanimate surface. So that's why they're called a slime layer, so they can stay on inanimate surfaces. That capsule allows them to do that. Flagella are an exterior organ of locomotion, so that makes them motile, made of protein filaments. They look kind of like a tail, so this is also a type of virulence factor. Organisms can be either monotrichous, which means that they have a single flagellum at one pole, so that's referred to as polar flagella. They can be lophotrichous, where they'll have 2 or more flagella at one end, so they're still polar, but they have more than one flagella at the end. Peritrichous is a uniform distribution of flagella all over the body. Amphitrichous, 1 flagella at each end. And then atrichous refers to organisms that do not have flagella. So absence of flagella, these bacteria would be non-motile. And let me go back up to this slide for just a second. So the capsule so this is an image of a gram stain as we would see it under the microscope. So this is the organism itself. So it's a gram-positive organism because it's purple. But you can see this kind of halo around the organism. That's what the capsule, that indicates that it's encapsulated, and that's what the capsule would look like on the gram stain. The next structure is pili. So these are non-motile, long, hollow protein tubes that connect 2 bacterial cells and mediate DNA exchange. So this is a type of virulence factor. Fimbriae are non-flagellar, sticky, hair-like appendages that allow some bacteria to adhere to one another and to environmental surfaces. So because of that, they are also a virulence factor. They're made of proteins. Spores they don't stain with gram stain either, but they're going to show up as opposed to the capsule, which shows up as an unstained area around the organism. Spores are going to show up as an unstained area within the bacterial cell. So they can be used as identifying characteristics depending on the location of the spore within the cell. Spores are kind of a big deal because they form during harsh conditions. They can lie dormant for many years until environmental conditions are favorable for the organism again. And then at that point, when they recognize that environmental conditions are favorable, they can emerge. So this is also considered a virulence factor. In this last little section of the Unit 1 Part 2 video, we're going to talk a little bit about bacterial growth and nutrition. So all bacteria have 3 major nutritional needs for growth. They have to have a source of carbon, they have to have a source of nitrogen, and they have to have a source of energy. So based on how these needs are met, bacteria are classified into 2 different groups. So they can either be autotrophs, which means that they are free living in the environment, so they're referred to as self-feeders. They are capable of making their own food because they can take inorganic CO2 and convert it to organic compounds, so they are not dependent on other living things for nutrition. Or they can be heterotrophs. These are our medically important bacteria. All bacteria that inhabit the human body fall into this category. They have more complex growth requirements, so they have to get their carbon from preformed organic matter, such as glucose, for energy. So there are also some additional environmental factors that must be considered when bacteria are cultured in the laboratory. So they have to have some type of source of water or moisture to help them from drying out, a source of inorganic salts, so that helps them regulate their osmotic pressure. pH, most bacteria are going to grow in the range of 6.5 to 7.5. Most culture media is around 7 to 7.5. They're going to have atmospheric requirements, so all require carbon dioxide, so that's what determines autotroph versus heterotroph. Obligate aerobes, so these are organisms that require regular air with oxygen for growth. Obligate anaerobes cannot grow in regular oxygenated air, so they're going to require special growth environment without oxygen. Aerotolerant anaerobes, they do not utilize oxygen but can survive and grow to a limited extent in its presence. So they're not harmed by oxygen. They don't use oxygen for metabolism. They just kind of hang out there. Facultative anaerobes do not require oxygen, though if it is present, the organism can use it and can grow faster and better in its presence. Microaerophilic bacteria can tolerate only very small amounts of oxygen, usually less than 5%. And then capnophilic bacteria, those are ones that like increased CO2 for optimal growth, so usually somewhere between 5 to 10%. We also have to take into consideration temperature, and that growth temperature that the bacteria are growing at can be diagnostic. So thermophilic organisms like very hot temperatures, so their optimal growth temperature is between 50 to 60 degrees Celsius, which is 122 to 140 degrees Fahrenheit. Mesophilic organisms like body temperature, so their optimal range for growth is 20 to 40 degrees Celsius, which is 68 to 104 degrees Fahrenheit. And then psychrophilic organisms like cold temperatures, so their optimal is 10 to 20 degrees Celsius, which is about 50 to 68 degrees Fahrenheit. We also have to take into consideration effects of neighboring organisms. So if there is more than one organism growing on the media. Colonization is growth of microbiota in or on a body site without the production of damage or notable systems. So after birth, microorganisms will find their area on the body that they wish to colonize, and then this is what becomes our normal flora, but our normal flora can change over time also. Symbiosis or mutualism, this is when both organisms help each other, so the effects are greater in a group, so they're working together to survive. An example of this would be lactobacillus in the urogenital tract of women. It prevents commensalism of pathogenic bacteria, and they obtain the nutrients that they need. Parasitism is when the organism benefits at the expense of the host. Commensalism, the organism benefits from the host without causing the host harm or without providing benefit to the host. So an example of that might be some of the organisms that make up our gut flora in humans. Antagonism are organisms competing for the same nutrients in an environment. So they're trying to fight one another off so that they can take over and make use of the nutrients. And then synergism, you've got groups of organisms cooperate to produce a result that neither of them could carry out alone, but that relationship is not necessary for their survival. Growth phases for organisms. So, and again, you may have seen this or covered this in maybe a previous biology course. So the lag phase, this is when bacteria are grounding themselves in the media. So they're just getting used to their surroundings. They're getting ready to divide. The log phase is when bacteria start to multiply. They multiply by binary fission, so they split in two. And this can occur as often as every 20 minutes, and it will continue as long as organisms have nutrients and their environment is favorable. Next is the stationary phase, where nutrients become limited, and even though the numbers of bacteria may remain the same, viability may decrease. So the growth rate will decline due to those limited nutrients and pollutants that are being excreted by the bacteria into the media. And then the last phase is death or decline, where bacteria will eventually run out of the nutrients and they will die. Okay, so a few little review slides here, and then we'll finish out this part 2 of the Unit 1 lecture. So identify the most fundamental test used for classifying bacteria. And we said that is the Graham's stain. What's the primary stain used in the Graham stain procedure? So we talked about all of these reagents. Which one is the primary stain? That is the crystal violet. That's the one that turns the organism purple. Okay, if you're looking at this gram stain result under your microscope, how would you report it? Gram-negative rods, gram-negative cocci, gram-positive rods, or gram-negative cocci? It would be reported as gram-negative rods because it's gram-negative. It has held on to the secondary stain, which is safranin, and it's a rod-shaped organism. Okay, this last little bit in this portion of the lecture is talking about morphological variations. So due to stressful conditions, the bacteria are not able to replicate in their normal fashion. So this can produce what's known as involution forms, or just you're getting strange results in your testing that can be caused by a few different things. So one of those things that can cause those involution forms is antibiotics. So if you think about what antibiotics are used for, they're trying to kill the bacteria. So bacteria may react or respond to those antibiotics and give you weird results in your testing. Inadequate growth requirements. So if the bacteria don't have all of those growth requirements that we talked about previously. Genetic alterations. So bacterial chromosomes contain all the information for cell growth and replication, but sometimes those messages get mixed up and can cause mutations. So plasmids are an extra little bit of circular piece of DNA that can get gained or lost. They're not essential for bacterial growth. They often carry virulence genes and antibiotic resistance genes. Conjugation is transfer of genetic information from one bacteria to another. They can pass on antibacterial resistance. So if one figures out or develops a method for resisting a certain antibiotic, he can tell his friends about it. And, hey, look at what I figured out. I've figured out how to resist this antibiotic. Let me show you how to do that also. And then there's also several other methods of genetic mutations that can occur that the book talks about. We won't really go into those. And then some changes in colony characteristics, so the way the organisms look on the plate. Just be aware that colony morphology can begin to change as these mutations and genetic alterations are taking place. So if you're expecting an organism to look a certain way on the media, but then you start to see changes in that, it could be because of some of these involution forms. So this is the end of part 2 to the Unit 1 lecture, so we'll pick up in the next video.